In this instructional video, we're going to show how to use the filtering mechanisms in the protein family sorter to be able to narrow down and examine a specific group or a specific group of features. So where we left off, we had launched the protein family sorter tool, and it had given us this table that has the filter on the right and the tabular results on the left. We talked about how you could get the pangenome here, the core genome here, and be able to identify protein families unique to individual genomes by clicking here. What I want to show you now is how to use this filter to get more info, to narrow down the results page to be more specific. Let's say you wanted to find all the genes or protein families that have the word flagella in them. So I'd have to go here, click filter, and you notice now we have 34. So these are all the 34 protein families. However, if I went flagellum, let's see if they have something different. Whoops, I have to click filter here. So see, you have to know what the things are named sometimes to be able to narrow down on these protein families. So the names, when we talked earlier about genome unique identifiers, there's also the protein family unique identifiers. Let me reset this to show all 5,000 results. A really good example of this is taken from this paper by this group of researchers that were looking at a Brucella isolate from a particular frog. And they listed all the genes within the paper that were involved in creating the O antigen, which is an important defense mechanism of the Brucella. I went through and I captured all the protein families that matched those genes. So let's go into PATRIC, click on workspaces, click on public workspaces, go to PATRIC workshop, click on protein family sorter, and where it says Brucella LPS genes, click on that row, and then click on download. When that pops up, it gives us, this was an earlier one, it gives us a list of the protein families that are involved in creating the O antigen in Brucella. So I think there are 72 of them. I'm going to copy all of those. Let's go back to the protein family sorter, and this is an awesome way to use this filter box. So you can click there and have those protein family IDs, and then I click filter, and it shows me just those 72 results. I wanted to show you another fun thing that you can do from the protein families. Here is, for example, let's choose one of these protein families. How about this one? I click on that protein family and it populates the vertical green bar with downstream actions. Let's click on multiple sequence alignment. This will open up a new tab and it will present an alignment and a gene tree. What we have across the top is something called web logo that summarizes the data down below, and it's bigger, like this is taller because it's showing that the number of genes that have methionine here. So it's bigger, and when it's totally conserved, it's small, and when it's very few have it, it's a smaller size. So you can scroll down the alignment, you can see areas like all of the melitenses, it looks like have a T here instead of an I. And so this is a really nice and fun functionality in PATRIC to be able to see these different protein families this way. Oh, look here. Here we have protein family. It looks like there was a frame shift because everything was going well in this Canis genome. And then it's there's a different sequence here, which is an indication that this gene is broken. In Brucella canis, this is a very important gene. When the Brucella have a functioning O antigen, they have a smooth phenotype when they're plated out. So they're identified as smooth. This particular mutation gives all the Canis genomes a rough phenotype, and it makes them different from all the other Brucella. So let's unclick this now. Let's reset the filter. And there are a couple more things to discuss. In PATRIC, you can filter on perfect families, which is one protein per genome. Note that right now there are 5,097 protein families. If I click on perfect and filter, it goes down to 3,999. So like a significant portion of the Brucella, their genes are very tightly conserved. You can click on non-perfect families and filter on that. And so there are 1,098, and the details of the number of proteins and the number of genomes that have them are here. Now, a non-perfect family may just mean that it's got 2 copies of that gene, but it could be that they're truly unique to it. So you could click on this and filter down and see which only single genomes have or a couple of genomes have. Let's refilter, reset that filter. Another thing you could do is I could say I want to see the number of proteins per family. I want to see all of the, I want to see the ones that have 10 to, how many are there here? 28. I don't know why I'd want to see that, but let's see. So you can do that. And I can also say I want to see 1 to 37 or any other number, filter that, and be able to see all the protein families that are not shared across everyone. And that's the way to use the tabular results to filter the information, and you can also launch a multiple sequence alignment from this. In the next instructional video, we'll be talking about looking at the heat map. So you're going to want to watch that. Thanks for listening, and thank you for using PATRIC.